Topic: Intro to Muscle Contraction. Assuming that you have studied the organization of skeletal muscles, we will now examine the steps involved in muscle contraction. Muscle action can be divided into four distinct phases: excitation, excitation-contraction coupling, contraction, and relaxation. Topic: Muscle contraction, excitation. Muscle intervention begins at the neuromuscular junction. Here, nerve action potential, NAP, moves down the motor neuron towards the synaptic knob. At this point, it is important to note that in biology, many body functions are based on electrochemical responses. Hence, electrical actions can cause chemical actions and vice versa. Therefore, an electrical action potential can cause voltage-gated calcium channels located at the synaptic knob to open and allow for calcium ion influx. The act of calcium influx allows for synaptic vesicles to release a neurotransmitter called acetylcholine, ACH, into the synaptic cleft. ACH, upon crossing the synaptic cleft, will bind to chemical-gated channels on the motor end plate of the sarcolemma. The ACH binding process induces the channel to open. Based on the electrochemical gradient, the opened channel forces sodium influx. Sodium ions enter the cell. Likewise, potassium outflux allows for potassium ions to leave the cell. Hence, the sodium influx forms depolarization, and potassium outflux induces repolarization. The rapid depolarization and quick repolarization forms a muscle action potential, MAP, on the sarcolemma. It should be noted that a threshold level of sodium influx must be met prior to the formation of an action potential. Topic: Muscle contraction, excitation-contraction coupling. Muscle action potential, MAP, formed on the sarcolemma migrates into the muscle fiber via T-tubules. Action potential, moving down the T-tubules, induces the opening of calcium ion channels within the sarcoplasmic reticulum. Calcium diffuses out of the SR and into the sarcoplasm. Calcium binds to troponin located on the thin filament. The joining of calcium to troponin causes a structural change that moves the tropomyosin belt and exposes the myosin-actin binding site on the thin filament. Topic: Muscle contraction, contraction. In the presence of ATP and myosin ATPase, an enzyme needed to activate the head into an energized position, the myosin head cranks into a binding position with the active site on actin. The binding process forms a cross bridge that connects the thin filament to the thick filament. In the contraction process, the myosin head pulls the thin filament towards the center of the sarcomere. In order to release the binding between the thin and thick filaments, a fresh new ATP is needed. The binding and release, also called power stroke cycle, continues in this manner. Hence, during the contraction phase, the thin filament slides across the thick filament and thereby shortening the distance between the ends of the sarcomere. The sarcomere shortens as the myofilaments overlap. Topic: Muscle contraction, relaxation. For relaxation, several steps are needed. First, the nerve action potential signal at the motor neuron must stop. Second, at the synaptic cleft, ACHE breaks down ACH. Here, the process of ACH dissociation occurs upon the formation of a muscle action potential on the sarcolemma. The synaptic cleft region is cleared of chemical action after the formation of MAP. This process ensures that only one contraction is created from one NAP and one MAP. Next, calcium ions are pumped via active transport back into the SR and held by a protein called calsequestrin. This process decreases calcium concentration in the sarcoplasm. As a result, calcium is removed from troponin. The final aspect involves the blocking of the myosin-actin binding site. Here, due to removal and decreased calcium in the sarcoplasm, troponin loses the calcium and allows for tropomyosin belt to return to the blocked state. Myosin can no longer bind to the active site on the thin filament. Result: the thin filament moves away from the center of the sarcomere, hence relaxation. Other filaments help with the return to optimal resting length.